Okay, so hi everybody. My name is Fraser Hof. I'm here to present to you at this edition of BioConference Live. Just by way of introducing myself, I'm a professor of chemistry at the University of Victoria. This is a university on the west coast of Canada. My area of research is in recognition chemistry, chemical biology, and most especially the chemical biology of epigenetics, methylation pathways, stem cell regulation, and cancers. So I've been asked to share a little bit about this story with you today, and I've also been asked to share with you that this talk is CME accredited, and so if you're after CME credits, then there will be a button on your screen at some point that you can use to follow up and obtain those. The title of my talk refers to the histone code and epigenetic methylation pathways and stem-like cancers. And I'm going to spend a little bit of time warming us all up to these particular topics. So the outline of my talk starts with really an introduction of epigenetics and the histone code. We'll talk about what histones are and how they work. We're going to talk about our own group's efforts to do reading of the histone code with a series of chemical sensors. We're going to talk about how methylation mechanisms are epigenetic mechanisms that control stem cells and cancer cell behavior. And finally, we're going to talk about a therapeutic approach that we're pursuing, which is chemical disruption of one particular member of these families. Now, this is a bit of a wild card for me. It's hard to know exactly who your audience is in these particular forums. You know, it's quite an interesting online session. I'm just going to ask a couple of poll questions here that I hope you'll all take the time to answer. And the first one really is just for you to categorize yourself somehow as one of these categories, so that I can really just have an idea of exactly what audience I'm talking to. Okay, so thanks for those answers. I'll just kind of pop the answers up here so you get a little bit of an idea. It looks like my audience today is mostly researchers, either on their way to a PhD or with a PhD. There's a medical doctor in the crowd. There are a bunch of people who have actually just joined in and didn't get to answer that poll, and so we're going to continue on the basis. Now, whether you're a researcher or a medical doctor, I'm also interested in knowing what your primary interest is when you see this title and you decide to attend this talk. So if you could answer this poll question for me, that would also be really helpful for me to shape the rest of the content. Okay, just waiting for a few more answers to roll in. Okay, so I won't even push these results back out to you, but considering that you're all researchers, I'll just share that the primary responses here were that you're interested in cancer research and epigenetics research, and that seems to follow nicely from your identification of yourselves all as researchers. So, okay, so here we go. Enough of the preamble. You know, the fact of our genes existing in the middle of our cells is something we learn from a very early age in science classes, and pictures like the one at the top left of this screen here, you know, our generic cartoon of a cell, are really routine, and you always see the dark patches in the middle, and we're told that this is the nucleus, and this is where the genetic material is, and it's called chromatin, and it's called chromatin because it refers to the dark color of that substance in microscopic images from very early days. Now, chromatin, in fact, is a packaging of DNA. You know, the actual DNA, the linear length of DNA in any one nucleus in your body is 6 feet long. So such that my hands wouldn't even fit on within the camera view here. So that 6 feet of DNA is packaged into your nuclei, just like string is wound around a kite spool. And the spool in this case are a family of proteins called histones. So histones have been known for many decades to be DNA packaging proteins, and you know, our understanding of their functions was really limited to just that simple function that they were packaging. Now, as many of you know as researchers, the world of histone biology has actually gotten very exciting in the last 10 or 15 years. And I'm going to talk to you a little bit about our interest in that today. Just to lay a little bit of groundwork here, there's an interesting thing about histones. You know, histones are proteins. When DNA replicates prior to cell division, you just stands to reason that you need twice as many histones to make up for the fact that you have twice as much DNA as the cell is preparing to actually divide and become two daughter cells. What that means is that histones are inherited. So DNA is inherited, and we all understand that, but histones are inherited too. And this brings us to a definition of epigenetics, which is, you know, loosely defined, that epigenetics refers to changes in gene regulation or gene expression state that don't involve changes in DNA sequence. And in the original definition, there's reference also made to the fact that these changes can be inherited. So everything I've told you so far implies the fact that histones then are inherited, that they're not DNA themselves, but they have obviously something to do with gene regulation. So this gave rise to the hypothesis, first a phrase coined in 2001 by Yenyuvine and Alice, that there was a histone code, and that the histone code was the means by which the readout of this code would be controlling gene regulation. So what I have cartooned here on this slide is again another representation of histones. We have here a sequence of a histone tail that's actually projecting out from the core. And what's shown here is a whole bunch of cartoony ways of representing that these histones actually are home to post-translational modifications. These are chemical modifications that actually are installed specifically onto different histone residues. So this gives us the metaphor of a histone code. And if we're going to follow that metaphor through to the end, you know, you'll see a list on the slide that we have methylation, acetylation, phosphorylation, et cetera. These are all different modifications that can be made. Those modifications then, in this metaphorical sense, are installed by enzymes that we call writers. So these are laying down marks. They must be read by proteins that actually bind to those modifications and have the downstream effect of turning a gene on or turning a gene off or doing some other regulatory event. And if we're going to follow the metaphor through to the bitter end, there are erasers. And these are enzymes that remove the side chain modifications again. And in my talk today, I'm going to talk to you briefly about a writer enzyme, just as an example. I'm going to talk to you about my own lab's interest in reading the histone code in various ways. The fact that these are gene regulation pathways really should tell all of us instantly that they have a high relevance to cancer. I mean, any kind of gene regulation pathway is co-opted by some form of cancer, and these are well understood when we talk about receptor tyrosine kinases. These are well understood. We talk about P53. All of these different systems, whether they're related to each other or not, if they are involved in gene regulation, there's the possibility they can be co-opted for driving abnormal gene regulation and therefore abnormal growth of cells. So just as an introduction, there are many, many epigenetic targets that are currently being investigated as possible sources for new therapeutics. I really do want to drive the point home here that this is early days in this family of approaches. So this is only a small selection of targets I have listed on this slide that are related to the histone code or these histone modifications, and that are considered drug targets at current time. So the solid red arrow in the middle of the slide refers to histone deacetylase, that's HDAC1, and that's actually a target for which actual drugs exist that are actually used in the clinic. They're FDA approved, and they're used in the management of different malignancies. But that's the only solid red arrow on the screen because there are so few approved epigenetic drugs and epigenetic drug targets that have actually been proven. The dashed arrows that point at DOT1L and EZH2, these are meant to indicate that these are proposed drug targets, and these are the only two agents among the ones listed on this slide that have actually had agents enter clinical trials. These are just phase 1 clinical trials, just started within the last 12 to 18 months, pioneered mostly by a drug company called Epizyme, whose name kind of says it all. You know, they're a company that believes strongly in the promise of these approaches. Besides that, what we really have here is a huge landscape of promising targets and not so much a lot of validated targets. So these histone modifying enzymes on this small list are at this point validated drug targets that are showing responses in human beings, and the whole rest of this landscape is really not yet. So let's just really lay the last little piece of groundwork here, and we're going to talk about methylation, and we're going to talk about lysines. Okay, so I have pictured here a lysine side chain, and I just want to point out that when we talk about methylation as a modification, one thing very interesting to chemists in this family of modifications is that the addition of 1 or 2 or 3 methyl groups is really a subtle modification. I mean, not only is the addition of a single CH3 group the most subtle possible modification that one can imagine in terms of making a new covalent bond. It's the smallest modification in all of biology. But there's another important feature here, is that when a methyl group is added 1 or 2 or 3 times, the charge on that lysine side chain doesn't change. The pKa, the ionization state, is hardly perturbed, and so this is really unlike phosphorylation. When an alcohol group is phosphorylated by a kinase, you have a bulk change to negatively charged state that has very big influence on binding partners and on the structure of the protein that's phosphorylated. And these are much more subtle modifications. And one of the fundamental interests is how such a small modification can have such a big effect when you think back to this original picture that I showed you of chromatin. So we have here kind of a series of exploded views of chromatin, and you know, the methylations, just to show in cartoon form here, methylations might occur on a single lysine residue that is on the tail of a histone that's protruding from this massive complex. And even just a single site being methylated can have dramatic consequences for the actual expression of the genetic information. I'll give some examples of that as we move forward. So I feel like we're all on the same page at this point. I'm going to move on to talk about the idea that we can use chemical sensors to actually read the histone code. And really what we're talking about here is a new kind of analytical approach. So I'll introduce you to a family of molecules. There's one very simple example pictured in the top right corner. I'll have another picture coming up soon. These molecules are actually called Cullix arenes. And they're relatively compact. They're macrocyclic small molecules. And what we found early in our research group is that these particular molecules, although we never invented them, what we found is that they're really beautifully suited to binding methylated residues. And there's a picture on the top right of the screen here that kind of implies something about the three-dimensional structure of the molecule. They're called Cullix because that's from the Latin for chalice or cup-shaped. And so these molecules are actually nicely a cup-shaped molecule. And if you decorate them with the right chemical functionality, then you make a cup that is actually able to bind other elements within it, within that binding pocket. So there's a picture shown on the screen now, and the green molecule is a simple Cullix arene. And the orange stick structure here is actually a piece of histone. And it's a piece of histone tail that has a trimethyl lysine on it. And what you can see is that the methyl groups on the lysine are buried inside of this binding pocket. This model is not just a flight of fancy. This is really built on a lot of different experimental evidence. What we're essentially doing here is in a really fundamental way, we're carrying out a recognition event that constitutes reading that methylation state. And just to put some numbers, to go back and put some numbers to this, these molecules, calixarenes, have been well known to bind various positively charged bits and pieces of protein and bits and pieces of other molecules, but they have a really special, really perfect complementarity to the structure of trimethylysine side chain. And you can see that in the affinities here. Essentially, this simple molecule has an affinity for trimethylysine that is as good as many of the proteins that have evolved to do the same recognition the same recognition task. And in fact, it has selectivities not quite as good as the evolved proteins, but still very reasonable selectivities, 70-fold selectivities over unmodified proteins. We can get that up over 100-fold with different modifications. Easy selectivity over positively charged arginine residues. I mean, there's a fortuitous complementarity here in this family of structures. So how might one use this kind of structure to do an analysis, right? So let's consider the analysis tasks that are required to begin with. The reason that the histone modifications are called a histone code is because of the enormous complexity and the enormous diversity of the number of modifications. So I alluded before to the fact that both methylation and phosphorylation and acetylation can happen. And what one must consider to understand the complexity of this is that all of those things can happen within a very small structural motif on a histone. They can all happen at the same time, or they can happen differentially. And the actual number of permutations and combinations, when you consider all of the known modifications on a single histone, are really staggering. So one can very quickly do a kind of back of the envelope calculation and facetiously find that just considering the number of different known modifications that exist on a single histone, on histone 3, the total number of different permutations and combinations is far more than the number of stars in the universe. Okay, and the reason I call that facetious is that there's no particular reason to think that that number of different modification states is relevant in biology, but we do know that there are definitely complexities here that make analysis challenging. So as these analyses are done right now, every one of these analytes, whether it contains 1 or 2 modifications or maybe 3 modifications, they require one single antibody. And of course, antibodies are incredibly powerful tools. The problem is that they really don't work very well on histone tails. And there's a lot of literature. That tells us explicitly, quantitatively, how bad the antibodies are. So I had a question just come in there, and I'm going to field that question about calixarenes. So just one more moment when we get back there. But thanks for that question, and I'll point out to all of you that you can definitely write in questions at any point. And I'll definitely field them as they come in. So the way that antibodies work is that every one analyte that one might be interested in requires a different antibody. Now, it's a bad idea as a chemist to set out on the chaotic quest of preparing the perfect supramolecular receptor for every possible histone-based guest or every single possible modification. The state of chemistry is simply not good enough to do that. So really, when we talk about lock and key recognition, and my starting point here is that there is no such thing as the perfect lock for every single one of these keys. So we need a different approach. And we take our inspiration here from a completely different part of nature. We take our inspiration here from a pattern recognition kind of analysis that happens in your nose, happens in many other parts of nature too. The nose is a great example. Every human nose contains 391 different olfactory receptors. When you smell a certain molecule, so for example, camphor, this is a natural product. It has a very characteristic smell. It's the smell of Vicks Vapor Rub, if any of you are familiar with that product. So when camphor binds in your nose, it doesn't elicit a smell response in your brain because there is a camphor receptor. There are actually a family of receptors that sense that particular family of smell compounds. They produce a pattern of signals that are sent to your brain. And your brain then can analyze that pattern, and it can determine that that pattern is the smell of camphor. And in this way, we have 391 different receptors, but most humans can easily identify and separate over 10,000 different smells. And so it's this pattern-based processing that allows us to tell many things apart from few data points. And there are various chemists listed here who have pioneered making chemical sensor arrays that can kind of reproduce this feat. So our first step then, if we want to do this with our macrocycles, is that we really need to make the macrocycle binding event something that we can get a readout from. And the simplest way to do this here is actually to use a fluorescent dye as a proxy that tells us about binding. So I have a scheme here that shows what's called an indicator displacement assay. And I have pictures of a particular dye and a particular host and a trimethyl-lysine in this example. And just to show you quickly, when you add the analyte, a trimethyl-lysine, you compete and you outcompete the dye. Some dye is released into solution, and that dye therefore gives you a numerical response. How intense is the response is really simply how bright has your solution become. Excuse me. And the way that this looks in a 96-well plate is like this. You can see running vertically here, we have different amounts of brightness in a particular row of wells, and that just corresponds to the actual visible response we get from adding a piece of histone to this particular series of wells. So we've made sensors, and now we need to make an array of sensors. If we want a pattern of signals, then we really need to be able to generate different data from a given analyte. And so the way that we did this was to take a very simple approach, and we simply expanded our toolkit very slightly. We instead of using the one parent calixarine that I showed you before, we added two other calixarines. And these things have each of them the ability to bind the dye, but each of them a different ability to bind different histone modifications. And I really want to drive the point home here that we did not program these things in order to give a certain pattern. We didn't program them to be selective for one modification or another. The idea behind this approach is really that you don't need to program recognition. You can add an analyte to your array of sensors, in this case 3 different sensors, and you can get a response. So for example, for arginine, the letter R on the graph at the top right, we get a pattern labeled A, B, and C from sensors A, B, and C. You can see that the patterns here are different for each particular analyte that we add. And using some very simple mathematical processing, we can convert those patterns into a more robust way of looking at the data. For those of you interested in the details, this is available in the paper. If you follow that reference that's at the bottom, we use methods called principal component analysis or linear discriminant analysis. And these really, they're complicated to explain, but they're very simple to execute, and they're routinely used in diagnostics science and in environmental monitoring and many other applied fields of science. But the point is that when we do these measurements many times, what we're able to do is generate data distributions that tell us definitively, once we've trained our set of data, what we're looking at. And I'll show you a couple of interesting examples here before moving on to the next topic. One can imagine that we tackle this problem of many and different modifications on a particular chunk of histone. There's an example here on this slide where we have an acetylated piece, an unmodified piece, a trimethylated piece, and a piece that is both trimethylated and phosphorylated. These things are very easily discriminated from each other by this method, even though the method isn't designed to recognize any one of those things. And we can get even more different. We can take trimethyl-lysine, or I should say more subtle. We can take trimethyl-lysine, and we can basically look at analytes that represent chunks of trimethyl-lysine on different pieces of histone. So you can recognize here that the only difference is in the sequence of the backbone. These are much more subtle differences, and some of these sequences are even very similar, and yet a simple sensor array here made of only these 2 molecules can tell these things apart. And moving forward quickly, I just kind of going to throw everything out there for you right now to show that we can take essentially any kind of histone modification state and train our sensor array on the original output, and then probe with a blind analyte or with a blind unknown sample and determine exactly where these things are. On the far left of the screen, you can see one example where these ovals, these confidence intervals are overlapping, and that happens only when we've made analytes that essentially don't bind at all to our sensors. And here and there throughout the screen, you can see things that are really very closely related and yet easily discriminated by this method. So that's one kind of biochemical analysis. If you think about what this might be used for, there are definitely diagnostic applications we're pursuing. Something we're pursuing even more closely is the use of these things as enzyme assays, because we get a very rich set of information out of these things. And the red dotted arrow that I've just made appear on the screen really represents the reaction of a demethylase enzyme. If you look at this very small text, we have H3K9Me3. Those data arrive in one sector. And when we remove the methyl groups by the process of an enzyme, which is a demethylase enzyme selective for that substrate, then we can very robustly read that enzyme reaction out in a really information-rich way. And that's something we're very interested in pursuing as research tools. We have both diagnostic applications and research tools that we're developing based on this fundamental technology. And I recognize that many of you in the audience are probably biochemists or cell biologists, and so I would be remiss if I didn't mention that these reagents are actually also really well suited for doing pull downs and interfering with pull downs in a variety of ways. So pull downs are really a bread and butter tool for many biological researchers in these areas. And so this is why we headed in this direction with the help of collaborators at the University of Colorado. You know, we're able to show that our molecules are effective enough at doing their job, whatever it is we've programmed them to do. In this example, to bind a methylated histone tail, that they can robustly interfere with pull downs. And in other arrangements where we actually have the calixarenes themselves on solid support, they can do the pull downs. They really work very well with this whole mode of experimentation. And that plays nicely for fundamental studies of interactions among biomolecules, and also using these things as reagents leading into proteomic studies. So there's something I want to make very clear here. These macrocycles, although they bind to methylated histones that are related to disease states, we really don't pursue the development of these things as drugs. And that's simply because they're fundamentally not drug-like, and we think it would be a bad idea to spend a lot of effort trying to get them to be drugs. With that said, they can have impacts in cells, and we've shown that they can get into cells. We just feel they just don't have the right physicochemical properties or pharmacological properties in order to really be explored as reagents, as therapeutics in any realistic way. But as reagents, they promise to be powerful tools for a variety of things. So I'm just going to return to a question that I had earlier about the calixarene, and I'll return to it here because I have a nice picture in the top right corner. The question was, what are the features of the calix macrocycle and methylated lysine that are responsible for the good affinity? And the reason I waited until I had this picture come back up to answer that question is because it's really nicely shown in the top right corner here and on the left side. The important features in order, probably in order, are electrostatic attraction between the sulfonate, which is negatively charged, and the side chain, which is positively charged. That explains our basic level of affinity for any kind of lysine. When the methylation happens on the lysine side chain, that side chain becomes slightly bigger, it fits the pocket better, it becomes much more hydrophobic, and if you look inside of that green pocket from this top-down view, you can see that you have there a big patch of hydrophobic surface area, and so that very much helps to drive the binding. And we've done various control experiments to know that that's true, that hydrophobicity is very important. And finally, the most canonical of these interactions that most people would have seen first is actually the cation-pi interaction. I'll actually have a diagram of the cation-pi interaction a little later in the talk. But basically, the idea is that the pi electrons on these benzene rings that define the walls of the binding pocket have a partial negative charge that's very nicely complementing the partial positive charge on the methyl groups that bind inside of it. Okay, so I think that question is answered, and I do encourage the rest of you to take cracks at me with questions at any point going forward. So, okay, let's talk about cancer a little bit more directly. There are many examples of histone methylation being involved in cancer pathways, and I can't possibly cover them all here, so I'm going to give you one quick example of two related families. And in these families, what we're talking about are methylation events that happen to drive the silencing of tumor suppressor genes. And you can imagine if you silence a tumor suppressor, then you're going to promote growth and you're going to promote cancer. So in this particular example, which is just one of hundreds of such examples, we're looking at a gene here. The gene is CDKN2A. That is a gene that encodes a tumor suppressor. I'm going to show you first the writer protein, EZH2. That's a methyltransferase enzyme. It methylates a histone site that is in the promoter region for this gene. And you can see in the bottom, of, again, my cartoon for methylation being installed. And so EZH2 is strongly correlated with the silencing of this gene, and therefore with a variety of aggressive and metastatic cancers. And so here I refer to some data from the company Epigenetics. They have methyltransferase inhibitors of a few different kinds, really interesting different chemotypes involved. The one shown here is an EZH2 inhibitor they reported on recently in PNAS. And this really is just shown because it's a nice demonstration of the concept, okay? In the cancerous cell in this particular study, the tumor suppressor CDKN2A is silenced because of the action of EZH2. The addition of the EZH2 inhibitor creates, let's say it blocks methylation, and they can show that definitively. The blocking of the methylation leads to restoration of expression of the tumor suppressor. And so then if you look at the graph on the right, what we have here are some tumor volume curves in a mouse model for this particular malignancy. And you can see here a dose-dependent response that's sustained over time, where treatment with the agent that inhibits EZH2 is actually very significantly knocking the cancer completely out. And so the paradigm here is that if you rescue the tumor suppressor, then you're treating the cancer. This particular agent is not the one that's in clinical trials right now, but the same company has a related agent that has the same mode of action that is in clinical trials right now, just in phase 1, showing some interesting results that they've been disclosing in small pieces. Okay, so that's an example of how epigenetic drugs might work. I'm going to talk next about another part of this exact same family of results, which is a methyl lysine reader protein called CBX7. So I'll go back to this cartoon. And what I didn't mention before is that if a methyl group is installed, it really doesn't do anything until something else binds to it. And that's the family of proteins we call reader proteins. And this is a family my group is very interested in. So the reader protein in this particular pathway that mates with this writer and this tumor suppressor is called CBX7. So it reads methyl groups. And this is actually just now a scattering of different pictures of complexes from different reader proteins reading methyl groups. And so what we have here are the gray structures are different reader proteins. And the green structure is a highlighted trimethylysine. And there's something interesting that you'll see here. These are very diverse and unrelated reader proteins, but they all look a lot like the calixarine. They all have aromatic residues arranged in a cup shape that surround the methylated side chain. And that binding motif is called sometimes the aromatic cage. And it takes advantage of the same interactions as did the calixarine. It has cation pi interactions depicted at the bottom center of this slide. It has van der Waals interactions that have to do with filling the space properly, and it very much relies on the hydrophobic effect in order to achieve the selectivity for the methylated state. And I'll point out here, although it's not on the slide, there are also electrostatic interactions that are important in that there are very often carboxylate side chains very close to the very close to the actual methylated side chain. So if I can just highlight these things here, there's an example, and there's another example. Those are negatively charged side chains in the reader protein that are very close in complementing, giving that minus attracts to positive kind of interaction also. Okay, I have another question coming in here. Oh, sorry, I just didn't quite state that clearly. The motif here is called the aromatic cage. And that's simply a reference to the fact that these aromatic residues are typically arranged so that they're each in a cage around the methylated side chain in different ways. Okay, so what does CBX7 do? What is its biology? Well, it turns out that CBX7 is very tightly involved in the regulation of stem cells, both embryonic stem cells and adult stem cells. So these are cells that can become lots of different things. We've done none of this work. You know, the work of biology comes before the chemists like me get involved. But I can just point to the work of Emily Bernstein, DeHaan, De Croce. These are like pioneers in understanding CBX biology and especially how it relates to stem cell regulation. So just to give you a very brief snapshot of that, in stem cells, CBX7 is silencing multiple cell cycle checkpoint proteins. In doing so, it has the specific outcome of maintaining pluripotency in stem cells and giving a general proliferative signal in stem cells. And if you find loss of CBX7 in a particular stem cell population, then what you have is actually the state of the stem cells becomes less stem-like. They differentiate and they become exhausted, or they just become other kinds of cells. They're not stem cells anymore. So stem cell is really a master regulator that makes a stem cell, or that gives cells stem-like properties. And so it's very easy then to imagine how it is that these features of CBX7 in normal stem cells can be co-opted in cancer. So I've already said that it silences multiple cell cycle checkpoint proteins. Of course, cell cycle checkpoint proteins are very often also called tumor suppressors. Upregulation of CBX7 has been identified in several cancers. It predicts poor outcome in several cancers, although I'll be clear here, not all cancers. The profiles are different in different tissues. If you knock down CBX7 expression, you strongly reduce proliferation in cultured cancer cells of a few different types, and stem cells of some types. And I'll just point out that very importantly for a medicinal chemist, these proliferative effects of CBX7 depend on CBX7 having a competent methyl lysine binding domain. And if you look at the actual curves that are shown here, the ones of interest are CBX7 wild type, which is the red curve, and CBX7 W35A. These particular authors in this paper, they used a construct where a single tryptophan that binds a methyl lysine was mutated away, and they showed that that completely shut down methyl lysine binding. And in fact, then what you get is this. So you don't have any of the proliferative effect that you normally see for normal CBX7-driven proliferation. So this is all related to cancer cell growth. It's also related very tightly to stem cells. And to sum up a lot of controversial literature, stem-like cancers are a subset of cancers that are aggressive and very hard to treat. And we think that CBX7 antagonists will halt proliferation in cancer stem cells and in other hard-to-treat cancers where CBX7 is a driver of aggression. Okay, so how do we do this? So we're going to back up and think back to the molecules, because this isn't a validated target. And in fact, no methyl reader protein is a validated drug target at this point. So we don't have kit assays that we can use. We need to develop our own assays. And our favorite assay in this family is called fluorescence polarization. Fluorescence polarization here simply takes advantage of the fact that we have a dye attached to a piece of histone, and the rate of tumbling of that dye gives us is a readout that we can actually very easily measure in any simple plate reader. And so this readout is called fluorescence polarization. You're certainly welcome to look that up if you want more details, but in a nutshell, In the presence of the complex, we have high signal, and when we disrupt that complex with a proper inhibitor, then we have signal go down. Okay, and just to show the example of many replicates, here's an example of many replicates of data where we have high signal for fluorescence polarization, and here's our positive control set where we have many very easily reproduced positive results, which is a decrease in fluorescence polarization. Okay, so our approach to do this was actually not high throughput screening initially. These targets are very hard to screen against. They have a lot of practical difficulties that make their high throughput screens difficult to succeed with. And so what we started with is actually the histone sequence that we know CBX7 binds. It binds to a certain methylated histone tail. And so we started with that. And now we're just going to run through the steps that a chemist takes when they're trying to start from a peptide, really start from a protein, and make something that is actually a useful tool for binding. And so the first step is to truncate that sequence to a minimal length. And so we started with a 14 amino acid long peptide. We found we could get as low as 5 amino acids long without losing too much affinity. The second step, also not shown in detail here, is that we really messed around with the sequence of the peptide at that point quite significantly so that we could get a better starting sequence than the native histone tail. So we still just have peptides here. Our third step was actually to show that the fluorescence polarization assay is reporting all of the structure-activity relationship that we want it to. And so here I have some red arrows showing these are simply IC50 values. Lower numbers means more potent. If we remove the methyl groups, then the affinity goes to immeasurably low. So we know that our peptide still relies on methylation in order to bind. Here's another example. If we actually make a modification here, that particular methyl group is this methyl group. It's buried in a cozy little pocket in the structure. And so we anticipated that if we made it any bigger, that it would destroy binding or completely remove binding. And sure enough, we identified that. Again, we have the predicted structure-activity relationship. If we make it a little bigger, we completely remove binding. A much more thorough accounting of all of this, by the way, is reported in our own paper in JMedChem, published earlier this year. Lots of steps that we can skip over for the purposes of this talk. So we have a crystal structure. It shows a really nicely closed binding site. And a closed binding site is good for expecting small molecule affinity of drug-like compounds. And to cut this long story very short, we basically were able to start with this 5 amino acid long peptide that's pictured on the left. And by making only 63 different compounds, almost all of these compounds were prepared by very simple solid phase synthetic steps. We're able to arrive at something, an example of which is shown on the right, which is a modified structure here, a peptidomimetic. The arrows here refer to things that are not peptide-like. We have a cyclopentane, we have a particular diol tail, we have a big aromatic group on the end. These things each were found to be very beneficial for binding. And so then by the time we've done this iterative optimization, we've gone from compounds that are about 30 micromolar in the original parent case down to compounds that are actually 200 nanomolar affinity. So we're now at affinities that are much stronger than the wild type interaction. We can show in many ways that they're able to outcompete the wild type interaction. We're able to do some nice studies that show that we're really quite selectively on target with these particular tool compounds. Okay, so I'll just say here that we do have results from these compounds that suggest their promise. We have a lot of unpublished results on their behavior in various different cancer cells, especially considering stem-like cancers as prime targets here. We have liability here in that these compounds are peptide-like, and so they don't have very good cell permeability. But we do have here the first example of any inhibitor of any particular methyl reader in this family. And just to kind of zoom back out and give you the context, There's a very nice series of figures here that are provided by the Structural Genomics Consortium in Toronto and in Oxford. So they have this beautiful online resource called ChromoHub, and it really tells you everything you want to know about chromatin-related proteins, but it especially tells the chemist what they want to know about what chemistry has been done on different proteins. And so what we have here is a series of families. What they have in common is that they're all methyl readers. So these are domains that bind to methyl lysine, like the CBX7 that I showed you. When we started this project, there were no inhibitors of any methyl readers in any family. While we worked on the project, there have been a nice example or two of inhibitors in other families. So MBT domains have one inhibitor for one prominent member of that family that's available. Another family of domains called PhDs. They have one family published out of the University of Wisconsin. The result on MBTs was a result of work at the University of North Carolina and the Structural Genomics Consortium themselves. And each of these came from essentially from screening results. But as I've mentioned, screening is quite challenging in these families, and so our peptide-driven approach here has given us compounds that are represented by the three arrows up here in the chromodomain family. And so these three arrows represent three different molecules that we've made that are inhibitors of different CBX proteins. The point of this slide is really to show how much of this landscape is untouched, more so than to highlight what has been done before. Each of these reader proteins has been correlated to really dramatic, or many of them have been tied to very dramatic impacts in stem cell regulation and in cancer cell regulation. And there's a lot of room for medicinal chemistry explorations in this area. And if there's any one take-home message, I would say that it's this. There is a lot of promise in this field, and there's a lot of room for work to be done. And what's needed is for chemists and other researchers to focus on making tool compounds in order to learn which of these things is worth pursuing and which of them is less worth pursuing, but really to take advantage of the therapeutic potential. So it remains for me only here to answer some questions. Before I do that, I'll just point out that I'm very grateful to various funders, many of which are charitable organizations. So those include things like a motorcycle rally called Ride to Live, Prostate Cancer Canada, the Movember people who are also prostate cancer fundraisers, Prostate Cancer Foundation of BC. A lot of this work in the second half of the talk was funded by those groups. And on the tools development side of things, the funding is primarily from NSERC and from Genome BC. The students who do this work, just to finish off last of all, are fantastic. We have incredibly high quality, highly motivated students here at our university. I'm grateful for them every day. And I just want to leave you with their happy, smiling faces here as a sign off before I take any questions. That's all I have to say. Please, if you have any questions, feel free to fire them away at me now. Okay, nothing imminent. A couple of questions answered during. So that's it. I'm going to sign off of this session. Oh, wait, a question just came in. The question says, for reader assays, ... Is there more question to follow? All right, actually things are hopping now. Let's see what I can do. Okay. One question for reader assays from ActiveX Biosciences. How about mass spec approaches for omics-wide readouts? So that's an awesome suggestion. The mass spec people, the proteomics community, I think have done an amazing job in the last 3 years at really refining how to do methylation-based analyses. If I can back up and talk about the calixarine type reagents that we're developing, one of our primary goals is to be able to make pull-down reagents out of calixarines that will facilitate proteomics-wide analyses. So, consider them as a pre-enrichment step on a digested sample before going into mass spec, so that you can have many more methylated, interesting targets in the actual soup that you're trying to analyze when you're doing that LCMS-MS-based approach. There's a multiple reaction monitoring and MRM approach that allows you to see where residues have been methylated. None of those things have really hit the big time in terms of being used as diagnostics, but in terms of research developments, there are definitely things going on in that area. I have a question from Quanticell Pharmaceuticals. Is prostate cancer a potential indication for CBX7? We think the answer to that is yes, and other CBX7 proteins might be relevant here also. And we've been working quite closely with researchers who are prostate cancer specialists, and the short version of that story is that in castrate-resistant prostate cancer and in the transition from hormone-sensitive to hormone-independent prostate cancer, we see a role for CBX7. Now, whether you call that transition to hormone independence or the transition to castrate-resistant prostate cancer, whether you call those clinical markers bona fide stem cell-like transitions, I think is a very open question, and I wouldn't speculate whether they're the stem cell story is very tightly related there. But the general idea that CBX7 is correlated with aggression seems to be solid enough to follow up on, and that's one of our approaches here. Any other questions? Okay, so thank you very much all for attending, hanging out till the end. I'm going to sign off now and thank the organizers also for this pretty cool opportunity. I hope you all got something out of it, and of course you can find me online if you want to send any more questions my way. Thanks so much, and bye.